Welcome to Constance YouTube. Today, I would like to share with you on this topic. How stable is the COVID-19 virus on the human skin and what shall I do? Since the outbreak of the COVID pandemic, people have been worrying. One of such worries is whether the virus will be on my clothes. How about my hair, my shoes, my skin, my money or coins? Many researchers are working on understanding this new coronavirus, SARS-CoV-2, the abbreviations of severe acute respiratory syndrome coronavirus 2, which is the virus that causes COVID-19. As of 19 October 2020, it has infected over 40 million people around the world. This is one of the recent studies that has been reported in news. They found that the COVID-19 virus can stay active on skin for 9 hours, but for flu, it is 1.8 hours. And it has been reported in the Chinese newspapers as well. Here also, in the Japan's time. So the purpose of this video is made in response to these questions raised after reading this news. How shall I sanitize my hands? What sanitizers should I use? The original paper that has been reported was originally published in Clinical Infectious Diseases by Oxford Academics on the 3rd October this year by a Japanese research group. The title is Survival of SARS-CoV-2 and Influenza Virus on the Human Skin. Importance of hand hygiene in COVID-19. And this is the reference. It is available for free online. If you are keen, please visit the website and you should be able to get the paper for free. This is the background for the study. It is unethical to recruit volunteers to test on these highly infectious and pathogenic agents. Pathogenic agents means agents that are causing diseases. And there are actually more than one strains of SARS-CoV-2, and there are limited skin samples collected from surgical procedures. The limitations became the motivation for the researchers to come out with a hypothesis and approach for them to study these viruses. So the scientists hypothesized that they can generate a human skin model using skins collected from dead bodies, which is called forensic autopsy specimens. Skin from such samples deteriorates slowly and can be used for skin grafting even a day after death. And they think that can be used for further testing in order to mimic the human in vivo condition. In vivo means what actually happens in a living organism. So in this case, they use human skin collected from forensic autopsy specimens to simulate the living conditions in human skin. And then they use the skin model to study the survival of the virus. So the approach they took was to generate a human skin model and then to verify that the human skin model is a working model for grafting the COVID virus. Influenza A was first used as a virus model on the human skin model generated, which I call the model A in these presentations. Here, hands of 6 subjects who provided informed consent were used to study the survival of influenza A virus on their skins too. Here, in this case, I label this as model B. If there is a correlation between the survival of influenza A virus in both the model A and B, then the model can be used for studying COVID-19 virus. This is what they were trying to do. They also study the survival of the viruses on other surfaces, such as stainless steel, glass, plastic surfaces, but the focus here is on the work of the human skin. So this is what actually was done. First, they collected the human skin from autopsy specimen, and then they grafted the influenza A virus in the model, which I call model A, and they evaluated the viral stability. At the same time, they also recruited human subjects and evaluated the survival of influenza A virus on their skins. And in this case, I call the model B. These were done in comparisons in order to establish that the model A is a suitable model for grafting the COVID-19 virus. Take note, this is the model that has been created from dead human skin, but this is the real-life human subject who had given their consent to do the test on their skin. So after establishing the model A as a suitable model for grafting SARS-CoV-2, they study the survival of influenza A virus and SARS-CoV-2 in model A with and without ethanol. Without ethanol is for them study the stability of the viruses, how long the viruses stay in the human skin, but the skin has been collected from autopsy specimen, not live human. For the part that when they were testing with ethanol, that is with a purpose to test how effective is this ethanol in sanitizing the human skin. This is what they did. They applied viruses on the skin in the model A and incubated for 30 minutes at 25 degrees Celsius at a relative humidity of 45 to 55% condition, and then they applied 80% ethanol and incubated for 15 seconds, and then they checked the survival of the viruses. They will check how much viruses remain after the applications of the mean results are presented here. With the help of the model B, the human subject skin model, model A was successfully constructed using skin collected from autopsy specimen. The model A was validated when they used influenza A virus. Basically, the skin model A was working for them. SARS-CoV-2 and influenza A virus were inactivated more rapidly on skin surfaces than on other surfaces such as borosilicate glass, stainless steel, and polystyrene plastics. Is this a good or bad news? At least we are told that the viruses can stay on these surfaces for some time, and we may need to sanitize them if we want to be very cautious. The survival time was significantly longer for SARS-CoV-2 than for influenza virus A. The SARS-CoV-2 virus survived 5 times longer than the influenza A virus, which is about 9 hours for the COVID-19 virus and 1.82 hours for the influenza virus. Influenza A virus on other surfaces was inactivated faster in mucus when compared to medium conditions, while SARS-CoV-2 shows similar stability in both the mucus and medium. The medium here is a culture medium that is commonly used to keep cells alive. The survival time was significantly longer for SARS-CoV-2 in mucus than for influenza A virus. The COVID-19 virus survived 11 hours in mucus, but for the influenza virus, it only survived about 1.7 hours. Both SARS-CoV-2 and influenza A virus in the mucus and medium on human skin were completely inactivated within 15 seconds by 80% ethanol treatment. The scientists recognize that there are 3 limitations in their work. First, they only use 1 strain of the COVID-19 virus and only 1 strain of the influenza A virus. The influenza strain was not a relevant clinical isolate. Second, they only use 3 skin samples from autopsy specimens and 3 mucus samples. Third, they recognized that they were using a validated model, not a live skin model. The human skin harvested from forensic autopsy was used to conduct the stability study of the viruses and to evaluate the effectiveness of the disinfecting process. So what are the conclusions? They concluded that SARS-CoV-2 may have a higher risk of contact transmission than influenza A virus because this SARS-CoV-2 is much more stable on this human skin model, which I call the model A, and they recommended that proper hand hygiene is important in preventing the spread of the virus. I think both of these conclusions are valid. In the beginning of the talk, I mentioned that I shall try to address these questions. How shall I sanitize my hands? What sanitizers should I use? Based on this recent scientific paper, it appears that 80% ethanol is effective to inactivate the COVID-19 causing virus in 15 seconds after applications. However, in real-life situation, one may not know how long the virus had stayed on one's skin. In this experiment, the viruses were applied on the skin model A for 30 minutes before applying the ethanol. To translate everything that have been found by this paper may pose challenges. As mentioned by the authors, at least 3 limitations are there, and we had just discussed them. There are a lot more different factors that are in place in real life that we are still trying to figure out. So, must I use 80% ethanol? I do not think so. There are other recommendations. A concentration of 70% ethanol is recommended by the WHO for disinfecting small surfaces. And I think that a simple way to keep the hands clean is to wash the hands frequently using soap and water. Since alcohol is flammable, we should limit its use as a surface disinfectant to small surface areas and use it in a well-ventilated space only. And as for hands, use it when you need. Since there are some safety concerns which is beyond the scope of the present study, we can discuss this in a future video. Other agents may be used for sanitizing as well, and that will be a topic in a future video. I hope this video is educational for you. Please share with your family members and friends if you find it helpful. Please subscribe too. Please stay safe and take care.